Hello my dear students, how are you all? Hope all of you are fine. So in the last video, I discussed about the prophase first of the meiosis first. Okay, in that we have seen that the prophase first was further divided into 5 subphases: liptotine, zygotine, pagotine, diplotine and diakinesis. Okay, now we have to start the next event of the meiosis first, that is metaphase first. Just we have to compare also the normal metaphase of mitosis and metaphase first of meiosis first. I think all of you know what was the event that took place in the metaphase of mitosis. In the metaphase of mitosis, all the chromosomes aligned at the equator of the cell to form equatorial plate or metaphase plate. And if you remember, I told you it is the metaphase stage when the morphology of the chromosome is studied, because that is a stage when the condensation of the chromatin reticulum is fully complete. So all the studies related to chromosomes, like its shape, size, means all the morphological characters are studied at the metaphase stage. But here we will see what will happen in the case of metaphase first. In metaphase first, what happened? One more thing you should know, that by the end of prophase first, that ended with the event of diakinesis, the chromosomes are fully condensed. The chromosomes are fully condensed. Okay, means in the case of metaphase first, the chromosomes will be fully condensed. And you know that in the case of prophase first, all the chromosomes has got paired together. That synapses took place. So here what happened? All the paired chromosomes, will align at the equator of the cell. So these are the centrioles, these are the spindle fiber. If you remember in the mitosis, I discussed about the structure that is called as kinetochore. So I told you that kinetochore provides a site of attachment for the chromosomes. Okay, so kinetochore provides a site of attachment of the chromosomes with the help of spindle fiber. Means they will be attached with the help of spindle fiber. At which point? Kinetochore. So here you can see the chromosomes are there in the form of pairs. So what the point you will say, that what is the characteristic event of the metaphase first? So all the homologous chromosomes, don't simply say the chromosomes, okay, because here the chromosomes are present in the form of homologous pair. That's why you will say all the homologous pair will align at the equator of the cell, okay, to form equatorial plate. Okay. This is the only difference, that in the case of metaphase, the chromosomes, all the chromosomes align at the equator of the cell, but here in the metaphase first of the meiosis first, all the homologous pair will align at the equator of the cell to form equatorial plate. If I ask you how many chromosomes are here in this condition, in the metaphase stage, how many chromosomes are there? You can see this is one chromosome, 2, 3, 4, and they are present in the form of pair. Therefore, total we can say 4 pairs or 8 chromosomes. So we can say 4 pair chromosomes are there or 8 chromosomes are present. Okay. Why I am saying? Because that we will use this data for the coming stages. We will see how the reduction takes place in the chromosome number. So this is all about the metaphase first. Then this will be followed by the anaphase first. anaphase first. What was the event that took place in the anaphase of mitosis? Do you remember? That during the anaphase of mitosis, centromere has splitted. The splitting of centromere took place to form two daughter chromosomes. But here what will happen? This is very interesting result phenomenon that we are going to see here. Actually, because of this reason only, the number of chromosomes will reduce to half in the meiosis. You see how? Do you think that here the splitting of centromere takes place? The splitting of centromere does not take place in the anaphase first. You can see this is the centromere which holds both the chromatids, but still they are intact. The centromere is still intact. There is no splitting of the centromere in the anaphase first of the meiosis first. Then whose splitting takes place? Here in the case of meiosis first, during the anaphase first, the homologous chromosomes had separated. So far we have finished the metaphase first. The chromosomes were still present in the form of pairs and all of them were arranged at the equatorial plate. But here, during the anaphase first, these chromosomes, the homologous chromosomes, they will separate. The homologous chromosomes will separate. There will be, very important, there will be no splitting of the centromere. Now what will happen in the telophase? You see, some picture you can see from here. As these chromosomes are moving towards this pole and these chromosomes are moving towards this pole, therefore the nuclei which will be formed here, in them the chromosomes will be, these chromosomes will be present in that nucleus. Similarly, the daughter nuclei which are going to be formed here, these chromosomes will form the composition of that particular nucleus. Here if I ask you, how many chromosomes will reach towards the opposite pole? 1, 2, 3, 4. 4 chromosomes will reach here at this pole and 4 chromosomes will reach here at this pole. At the metaphase first stage, how many chromosomes were there? 8. But what happened? When these two daughter nuclei will be formed, they will receive only 4 chromosomes. So whether the number of chromosomes will reduce to half or not? Obviously you can see the number of chromosomes will reduce to half. Okay. So that's why meiosis is called as reduction division. And now you can see that actual reduction takes place during which stage? Meiosis first or meiosis second? Obviously that actual reduction of chromosome takes place in the meiosis first. So that is our next stage. That is telophase first. Telophase first. So what happens in the telophase first? The chromosomes will reach towards the opposite pole. Even in some of the cells, they will become decondensed also, and the nuclear membrane will be reappeared. This is one daughter nuclei. This is another daughter nuclei. So two daughter nuclei are formed. Even in some of the cells, it is observed that the chromosomes they become decondensed. Okay. So you can see these two daughter cells which are formed, they contain how many chromosomes? They contain only 4 chromosomes. We started from how many chromosomes? 8 chromosomes. Okay. So they contains only 4 chromosomes. Means the number of chromosomes is reduced to half. Okay. Then What will happen after the telophase first? There will be cytokinesis. Means this cytoplasm will be divided. But this particular stage, you can see even though these two daughter nuclei are formed, but whether their division is complete or not, their division is not completed so far because they have just completed the meiosis first. They have just completed the meiosis first. And this condition of cell is called as dyad of cell. This condition of the cell in which two nuclei are present in a single cell, that is called as dyad of cell. Okay, so in the examination question is asked that when this dyad of cell is observed during the cell division. So this dyad of cell is observed during the telophase first of the meiosis first. So this dyad of cell is observed during the telophase first of meiosis first. Why they are called as dyad of cells? Because even though they have the two cells, dyad means two. They have the two cells, but these two cells are not fully divided. They are uncompletely divided because they have to now undergo for the meiosis second. That's why they are called as dyad of cell. Clear? Then by this telophase first we can say meiosis first also will be over. But if you remember I told you, immediately after the meiosis first, meiosis second will not start. There will be a small time gap between the meiosis first and meiosis second, that is called as interkinesis. That is called as interkinesis. But very important, I told you, during the interkinesis, there will be no duplication of the DNA. There will be no synthesis of the DNA. This is simply a small time gap between the two successive meiotic divisions, means meiosis first and meiosis second. This is just a time gap. Okay, and this value of this interkinesis may vary from cell to cell, but it will not be very much time taking. So it is a small time gap only. After that, again, these two cells which are formed after the meiosis first, they will undergo for the meiosis second independently. Okay, means this will undergo for the meiosis second independently, and this one also will undergo for the meiosis second independently. So how the event will take place in the meiosis second, that will start in the next video. So hope meiosis first should be clear to you. Okay, so in the next video I will start the meiosis second, and then I discuss about the meiosis second will be very simple. Then after the meiosis second, means in the same video I will discuss the significance of meiosis, and by that this chapter also will be over. Okay, so this is all for today. Thank you.